maybe now.
Yes, now we can see.
Yes? So you see molecular biology of nucleic acid lecture?
Yes, now we can
Oh,
see.
okay.
Yes.
So this is just this syllabus which it's supposed to be connected with each teaching course. And so you see the course title, Molecular Biology of Nucleic Acid, that this is the lecture. And this the important is that this is from our faculty full-time and who is teaching stuff. So this is me, Igor Konieczny. I should introduce myself. And this is going to be a lecture in the classroom. Unfortunately, it's not going to be in the classroom. It's a pity, but this is not in my jurisdiction about where the lecture takes place. So unfortunately, entire course is going to be in Teams. I don't know about the exam. Probably we'll learn later on about the exam. Is going to be on in internet or in the contact in the classroom. We will see. So we're supposed to have 30 hours. This is free ECTS credits. And the course is obligatory. So, and the language is of this instruction is also English, as I already mentioned. So what that means that the course is obligatory. So there is your obligation to attend the course. And I am not very restrictive in this, but I would not accept if someone would have less than 50% attendance. Okay, so that means that the person is actually would not be able to even to get into the point to the exam. If you will have less than 75%, then we will have to discuss. So the safest way is just to attend to the course with attendance at least 75%. Yes? Do you have questions?
Yeah.
Yes?
How will be the attendance checked?
Well, through the internet, I can just check your presence in the, yeah?
Oh, perfect, thank you.
Yeah, in the computer. This is the only way. I'm not going to do this in any other way. Teaching methods supposed to be translated. I think that I have not the newest version. So the teaching method is going to be online, Teams, okay, and it's going to be presentation, PowerPoint presentation. I would also try to discuss in details experiments or issues, and with some drawings as well, and demonstrations sometime. It's a pity that we cannot meet because that would be much easier, but I hope even with a camera I can just show you some models and this kind of things which I think are important. Okay, so this is just an assessment method. So it's going to be written form exam. However, I shouldn't say that this is going to be a written form because the last year I completely changed the form, and sometimes you do not necessarily need to write anything. Sometimes sometimes yes, sometimes not, but let's follow this syllabus and then we will discuss at the end the exam, okay? So the basic criteria for the evaluation, so understanding by the student of the complex processes connected with nucleic acids and their significance in biotechnology, knowledge concerning issues of molecular biology of nucleic acids presented during lectures and currently discussed in specialist literature, and the knowledge of English and specialist terminology, allowing students to understand the discussed issue. And so we have actually three parts. One is knowledge, second understanding, and the third is English skills. Okay, so especially with not just the grammar, but rather terminology and understanding of English in the speaking and written forms, understanding specific terminology connected with nucleic acids and biotechnology connected with this. So the exam also will have three parts, and I would say three different tasks, separate tasks, and you have to pass each of the tasks. Okay, so you need to get at least 50% from understanding, 50% from knowledge, and 50% from the English skills. Okay, and these three independent tasks, if that would be need, you will completed it in the through the internet. But I specifically use the term tasks because these are not necessarily going to be specific question or sometimes a question, but you may also need to perform something, create the model or create the drawing or to draw the scan of some process. So not necessarily just the question and answer, yes, no, but kind of more complex. And the last year I did actually individual questions to all students, and you perform like individual, each of you perform individual task. So actually there is no problem with the time. It was full time just to complete the task, and sometimes I think with the, I don't know, it was 3 hours or something, and actually everyone completed it in a shorter that it was specified. So you don't need to afraid. Okay, so sometimes you ask question about the English, so this English skills, okay? So that would be some task to verify if you understand and you know the correct terminology, and this is it. And so sometimes the questions are going to be in English, and sometimes the questions are going to be in Polish, and of course for the foreign students the questions are going to be only in English. How you should answer? You can answer in English or you can answer in Polish unless it is clearly specified. For example, answer in Polish or answer in English a particular question. Okay? So, do you have questions considering exam? Yes? No? No. Okay. So let's maybe move on this one. No, this one. This one. Okay, do you see this?
Yes.
Yes.
Okay, so we kind of discussed the syllabus, a little bit about goals. So goal is just what I read, understanding of issues connected with nucleic acid research and specifically related with biotechnology. Understanding, also one goal is just to have a knowledge and understanding, and the third goal is just to gain the skill, English skill, connected with the terminology and understanding of research connected with nucleic acid. acids. Okay, so this is just syllabus, this is goals and lecture, and also at the end we will have exam, but we discuss the exam as a separate part that is going to be at the end on internet or maybe in the classroom, and you will get particular tasks to perform. Also, we discussed the attendance that I would expect from you at least 75% attendance, and this 50% is absolute minimum just to be able to get into the exam. And this what you see is just the outline of an order of lecture. So we will start next week a little bit about the history of nucleic acids, about the discovery of nucleic acids, discovery of DNA, investigation, so on, this keystones, milestones of research on nucleic acids. Then we will move to initiation of DNA replication, and we will discuss, I think, 2 lectures about what is necessary to initiate DNA replication. Then we will move to pre-priming, helicases, so priming reaction, helicases, then replication, I should say DNA synthesis, then aspects of topology, and then transposition, and then repair, and then we will have an exam. And the major task, I don't know if this is going to be visible, what I would like to you, so I will try to launch this movie. Do you see this, or this is only me?
Yes, we can see.
Okay, so this is just the model of helicase, replicative helicase on the double-stranded DNA as a template. And at the end of the lecture, I would like you to understand and be able to describe all components of replication complex, how they are connected, how important are they, and what actually we see on this, of course, kind of artificial movie, but as you see, the helicases and other components of replication complex. I think that I would be very happy at the end that you would be able just to describe it to others as well, not only to me, but also to others, to family and I don't know, colleagues, how it is and how the replication DNA replication is complex, and how it happens, and what are the rules behind. We will discuss, as I mentioned, history of discoveries of nucleic acids. This is going to be next lecture. Also some art and social aspects of nucleic acids, and structure of nucleic acids, structure of complexes built on nucleic acids, and what is the current knowledge about this. Also evolutionary aspects of replication, and the structure of replication complexes and replication proteins, and also regulation and control, so different factors which are involved in the regulation. I also mentioned about the helicases, so the structure of the helicases and those enzymes as molecular motors responsible for the unwinding of a DNA, and how critical this unwinding of a DNA is for process of replication, but also for human being and animals' health, regulation of this process, and aspects of cell proliferation, how it is regulated. Then we will move to the chemistry of DNA synthesis, as I mentioned, also the role of particular components of replication complex, then topology, different structures which are created within the living cells, structures of DNA, and we also will discuss transposons and transposition reaction, and at the end, aspects of repair of DNA, direct repair, types of repair, and enzymes which are involved in those processes, and the concept of changing of polymerases for different kind of repair and de novo synthesis of nucleic acids. So that would be it. We'll kind of discuss the double helix in a very detailed manner during the lecture. Okay, so as I mentioned, we will start it next week. Important thing is if you all have received invitation, I guess yes, because otherwise it would be no one in here. And also, I guess you also received the link to the today's lecture, yes?
Yes.
Okay, so if you received the link to the invitation to the group, I will put into the calendar next lectures, so you will automatically get the information about the lecture. And I also, using this method, would connect with you if that would be something necessary. I don't know, some problems with a lecture, canceling of a lecture. I hope not, but if, then I will just try to send you the information through the channel. Okay. So, questions? Okay. I would try to convince you during the lectures to ask questions. And maybe this is very important because, you know, especially during this pandemic time when we are sitting in front of a computer and it's so difficult to communicate. So please remember that whatever question is fine. Of course, please do not ask about the weather because we can see the weather, but the questions connected with a lecture, or even if you don't understand, don't be shy and please just ask me to repeat something. I can also change to the Polish if that's going to be easier, or maybe make a short statement in Polish if something I think it's going to be more complicated, or I will have a problem just to send you the right message with using English. So please ask, and please do not afraid that that this is going to be negatively charged by me. Okay, so I see that there is no questions. So if not, then we are meeting next week and we will start just regular course and then discussions and hopefully questions, and we'll start the course next week at 12:15. Okay, so thank you very much, and see you next week. Thank you very much. Bye bye.
Thank you so much.
Yeah,
Bye.
goodbye.
Bye.
Thank you. Goodbye.
Goodbye.
Thank you. Bye.